There we go.
Yep, there we go.
That's better?
Yep.
Okay, perfect. Great. Thank you so much for that introduction, Sarah and Amanda, and for inviting me and organizing all of this. It's really a true pleasure to be talking to you guys today about this. We're going to go through, so I'm a clinical geneticist at UCLA. Majority of my time is spent seeing patients in clinical encounters. So this topic is something that I talk about daily with my patients and their families. And it's certainly a question that's come up in the ASXL community. So it's really my pleasure to pull this together for all of you, so you have a little bit of a resource and some understanding about the process that goes into understanding genetic testing results, because it's not always as straightforward as we think that it is. So we're going to start by just zooming all the way out to the basics and talking about DNA. So DNA is our instructions, and I like to think of it as a multiple volumes of a DNA instruction book that tells our body how to do everything it does. And our DNA is our instructions that ultimately makes the proteins in our body, and the proteins are what is every cell and every part of our body. And so this is sort of a basic picture just showing the different parts of the DNA. If you start at the end here with the nucleotides, those colored hexagon type shapes, those are the basic nucleotides, the basic building blocks of our DNA. And our DNA is so long that it has to be wound up really tightly and built into these chromosomes, and that's how our body stores it over time. So the nucleotides are like the letters in the words in our DNA instruction book. A gene, which we talk a lot about, ASXL 1, 2, and 3 being genes, are like the words, or even sometimes they're so long that they're like pages in our DNA instruction book. And then all of that gets wrapped around these proteins called histones and bound up really tightly and turned into our chromosomes. So that's sort of the structure of our DNA, and it lives inside the nucleus of all the cells in our body. But what DNA actually does is what we call the central dogma. So our body reads those letters in our DNA instruction book and converts it into something called RNA, which is sort of like an intermediate in the process, and then ultimately makes protein. And the protein is what goes out into the body and does all the things that we need to do as humans. But this is a really important process. And while we talk a lot about the genetic change in our DNA, we also talk about studying the RNA and the protein, because those are the things that actually have an effect on the body. So when we talk about genetic testing results, we typically don't talk much about RNA. It's kind of a silent step in the process, but we talk about the DNA and the protein. And so I'll talk you through how that looks. But also just zooming out and thinking about what types of genetic testing you or your loved one may have had. We'll spend some time focusing on the testing that will identify an ASXL change, but it is important to know the different types of testing. And sometimes when we ask families for testing, we get copies of other types of genetic testing, like karyotype or chromosomes or a microarray, or various types of DNA sequencing. So a karyotype or chromosome study is actually just looking at that DNA all when it's packaged up and wound really tight into our chromosomes. And so this is really the most zoomed out look that we do at our DNA. We can't identify ASXL changes on this. We are only able to identify if there's entire missing or extra chromosomes or large pieces that are missing or extra. And so the scientists actually will look at those bands and be able to identify if there's missing or extra bands and can report out on that level. But we can't do sequencing here. So we're not looking at the letters on the DNA page of our instruction books. If we zoom in a little bit further, we do a chromosomal microarray. And in a microarray, think of it again, we're still looking at our DNA, we're looking inside those chromosomes now. And this time we're looking at the level of sort of missing or extra pages or chapters inside our DNA instruction book. So again, we're still not at the level where we're looking at the letters on the page. So we're not sequencing genes or reading words. We're looking at the level of missing or extra pages and chapters. So this is, again, still a pretty zoomed out view. We can't typically identify ASXL changes here, we could find, and there are some people in our community who have a deletion or a duplication in an ASXL gene. So that may come up in the microarray depending on how big it is, but this is still not sequencing. So we're still not finding the letter differences in an ASXL gene through a microarray. So that brings us to DNA sequencing. DNA sequencing is the part where we actually look at the letters in our DNA, and now we're zooming in and we're looking at the words. So we're actually sequencing the genes and can identify genetic changes inside the ASXL genes. So again, this is sort of an example, and this picture here where it shows the G, C, A, and T here, this is actually the readout and how it actually looks when we do the sequencing. We just get different kind of color peaks for all the different letters, the A, T, G, and C. And based on that, we are looking for single letter changes or a few letters that are missing or extra that could change how the DNA word or gene is read. Okay. So there are several different sequencing studies that exist and that many of our families have had. So the most specific one would be a targeted familial variant testing. So this is the case where perhaps your child has been identified as having an ASXL1 change or variant, and then testing is done on parents or a sibling just for that specific letter change. So we don't sequence the whole word or gene, but instead focus in on just the letter change that the family member is known to have. We can also do single gene sequencing. So in this case would be if we were very suspicious that somebody had an ASXL1 related disorder and the geneticist or clinician ordered single gene sequencing where we just looked at the letters in that one ASXL gene. Another common presentation or common testing approach is a gene panel. So many labs have neurodevelopmental panels, for example, where they take 200 or more genes that are associated with neurodevelopmental conditions, and they put them all together on a panel, and that may include the ASXL genes. And so some people have sequencing done where the ASXL gene is one of many neurodevelopmental genes sequenced all at once. Most commonly these days, the way that things are moving is that people end up getting more broad testing instead of more focused testing. And whole exome sequencing is quite common. So many of our families have had exome sequencing. This is actually a test, even though we call it whole. It does not include all of the genome. It includes around 2% of the genome. We have over 20,000 genes or words in our genetic instruction book, but we still only know about 3 to 5, it's kind of creeping up, it may almost be up to 6,000 genes are associated with human diseases. So there's still quite a bit of the majority of the genes in our body we've never associated with human conditions. And a large part of our genome is intronic variants, which we'll talk about a little bit later, that we don't see on exome sequencing. So what we do in this testing is while we do sequence all of the genes, we sequence what they consider to be the active parts of the genes called the exons, and the introns, which are the in-between spaces, are not looked at. And so for that reason, that's why we end up with only about 2% of the genome. But much of the parts that we don't sequence, we actually don't know what to do with, and we don't have human conditions associated with them anyway. So overall, the yield for exome is about 25 to 40%, depending on the indication. And we often do this as a trio with parents as comparators for the person who's receiving the testing. The newest kid on the block is whole genome sequencing. So this includes the exome plus the rest of the genome. So this is kind of a classic picture of, it's not just a slice of the apple, but it's the entirety of the apple. We talk a lot about genome as being the new sort of frontier and the next step. And for many families who are undiagnosed, it is. But even still, because so much of that apple we don't understand, we have a long way to go before we start to increase the yield on testing. So even jumping from exome to genome At this point, it still only increases the yield by a couple percents. That will get better over time, but is still something we're working on. When it comes to our ASXL families who already have a diagnosis, none of this really matters because you've already found your answer. But for some of our families who have an uncertain result and are still left in limbo and asking questions, it's important to know that our genetic testing that we offer clinically still has a lot of holes and a lot of opportunities for us to understand things further. So it's always possible there's another diagnosis out there that our testing hasn't picked up yet. And this slide is to kind of demonstrate that variation in our genetic code is actually normal. So this is an example of a filtration process in an exome or a genome. This is one that I did in residency in my training. But as an example, this was an exome that was done with 5 individuals included. So the patient, their parents, and 2 unaffected siblings. And when we compared all 5 of those individuals' genetic information, we found over 135,000 variations amongst the family members. And so that's pretty typical. It's common to have well over 100,000 variations amongst a family because these are normal. So it's common for us to have changes. We're not all identical to each other for good reason. And so it's the lab's job to actually go through a pretty rigorous process to filter all of those changes out and try and identify which of these changes might be causing a condition and which are normal benign variations. So you can see here at each of the arrows, we use different techniques to filter out those variants and try and hone in on the ones that we think were most likely to cause this child's condition. So we start with quality control. Is there good coverage? Do we think that these are actual variations or are they artifacts of how the study is done? And so we're able to filter things down that way to 120,000 variants. We then remove things like non-coding variants, so things that are in the introns and other spaces that we don't really know how to interpret. And that usually drops things down quite significantly. So we're down to about 31,000. We remove genetic changes that don't change the protein. So if the DNA sequence doesn't actually affect the protein sequence, usually those are not disease causing. So we remove that and cut that in half to about 15,000. And then we look at control populations. So things like, is this genetic change present in people who don't have reported genetic conditions? And is it present in a lot of people? And if it is, it's much less likely to cause disease because all those people are reported to be healthy. And that usually significantly cuts down the number of variants as well. So that's in this family, cut it down to about 3,000. Then we oftentimes employ sort of the family inheritance, which is why we talk about doing this as a trio, or in this case as a pent. So we included siblings, and we're able to then look at what changes are inherited and what changes are new, and use that data to help us interpret which changes might have an impact. But as you can see here in this family, we still ended up with a good number of targets that we needed to investigate to try and understand the cause here. So again, this is just sort of an inside look at what we do on the lab side when we're doing something like exome or genome, and why there's lots of variability in results. And also just pointing out that it's normal for us to have variations, and it really is up to the labs to help us determine whether those variations are disease causing or just normal benign variations. So our labs use a variant call system that's listed here. This is from benign to pathogenic. And there are criteria that are specifically used to quantify each type of variant call. So if it's, for example, a variant that's been seen really commonly in the general population, the lab may call it benign. If it's likely benign, where there's maybe some information that maybe it could be disease causing, but mostly it seems benign, they'll call it likely benign. The category that causes us the most angst and frustration is certainly the uncertain category. So our variants of uncertain significance oftentimes gets abbreviated as a VUS, or VUS is the other word people will use to describe it. And that really is the in-between space where we can't make a call in either direction. And then likely pathogenic and pathogenic are those that are expected to be disease-causing changes. So this is a really busy slide and really complex, and we're not going to go through all of it. But this is the actual criteria that labs use to make a call on a variant, to decide whether it goes into this benign category or whether it is pathogenic. And so they use multiple tools to do that. The first being population data, which we've sort of talked a little bit about. They'll look to see, do other people in the general population have this change? If so, how many people have it? Is it common or is it rare? Have we see it at all? In rare neurodevelopmental conditions, we would not expect to be seeing a variant in the general population. They then also use computational and predictive data. So we can, now people talk about AI, and yes, a lot of it has become AI based, but we use prediction software to try and understand how a DNA change might impact the protein. And so there are specific tools that have been developed to help model the protein structure and look at where that change would be and help predict what the impact might be. If we have functional data, we'll try and use that, and that can be really powerful. So that's in the case where somebody's looked at that specific letter change before and determined that it does or doesn't have an impact on how the protein end product functions. And those are typically done in sort of research-based labs and published in the academic literature. Then we do segregation data. So that's what we talked about, the importance of having parents as comparators to try and understand if it came from an affected or unaffected parent. And then specifically, if it's new in the individual and not from either parent, that is what we call de novo and can often help kind of push towards the pathogenic side of things, given that it didn't come from either parent. So they list some other things there at the bottom, but that is really the most kind of useful data that we use. We will also look to see if the genetic change or is present in other populations besides humans. So if we will sometimes look to see, is it conserved across multiple different species? And that can be helpful information too. But none of these different pieces of information by themselves will make a variant call. It's really a combination of all of these things together that will determine what a lab will call as either being disease-causing or benign. And so there's been some work to understand why labs disagree, because it is true that if you send your DNA to 5 different labs in the US, you may get as many as 5 different results. And that has to do with the fact that the interpretation, even though we all use the same system, is very human-based, and not every lab interprets things exactly the same. So you can see here that the big circle in the middle is just that those criteria we just went through is that the labs apply them differently, and that is the biggest reason why labs disagree. There's a small chance of things like lab protocol issues or errors putting into it, but primarily it's really that interpretation part is done differently by each individual in each laboratory. Okay, so let's jump in. And we talked a lot about kind of how these different variants get called, the process, the genetic testing. We're going to jump in and talk a little bit about what a sample lab report actually looks like. So this is an example of a whole exome result, and many of the labs look quite similar. I couldn't use anybody's personal information here, so this is not an ASXL result. This is an example that's directly from the lab's website, but this is pretty consistent with what most exome reports look like. And so right next to the green arrow there, you can see the summary of the primary finding in this individual. And in this case, the gene that they found a genetic change in is MPZ. And what you can see there is the address of where that genetic change happened. So the C dot stands for coding DNA, and C dot 532 del is the address in the DNA. So at position 532 in the MPZ gene, there is a deletion. Okay, so the nucleotide there is deleted in the DNA. What follows is the protein. So the protein position, or the P dot. And in this case, there's a genetic change at position 178, where instead of a valine, there's a tryptophan. And when that happens, it results in a shift in how the protein is read. And after 74 amino acids, which are the building blocks of the protein is shorter than it should be. Okay, so the way that the lab writes that is that P dot phrase there, basically showing that the DNA changed, caused a change in the protein, which caused the protein to be shorter than it should be. And so this has resulted in what the lab is calling a likely pathogenic frame shift truncating variant. Frame shift because when this specific deletion happened, it shifted how the protein amino acids were read, and then it truncated it. It made it shorter in the end. Okay. So I tried to write here some of the abbreviations you might find in your family's reports. So sometimes amino acids we write as 3 letters. Sometimes we write them as a single letter. So you may see them just as single letters or 3 letters. The sign for shortening it can be an asterisk, and it can also be an X. And so you may see on your report an asterisk or an X there as well. Okay. So there's a couple different ways to write it. But again, what you're looking at is your C dot is your DNA sequence and where that changes, and your P dot is your protein and where the change in the protein occurs. The labs also will typically have on the second or even third page some information about the specific letter change that's being reported. So it's very common to have a paragraph or 2 like this, where the lab will describe the information on the variant call. And so this is where they'll go through a lot of the criteria we just talked about, where they'll explain why they decided that this is a likely pathogenic change. And then they will also provide a paragraph that talks about the clinical correlation, and so what the disease is and what's been reported about it. So this is a pretty common occurrence in most lab reports. In this person's exome sequencing, they also had a secondary finding, and just so we can kind of go through it again and look at a different variant, because I know it's confusing. This individual also had a change in a gene that caused an increased risk for certain types of cancer. And so they have a different genetic change. So going through it again, this gene is PMS2, and the C dot, or the coding DNA, at position 1297, there should be an A, and that greater than sign is showing that it's a T instead of an A. And so that change there results in a change in the protein that changes a lysine at position 433 to the end, an X or an asterisk. So it shortens the protein right there where that letter change happened. Okay. And so this is, again, a likely pathogenic truncating variant. And many of the members in our community, in the ASXL community, have variants that look much like this, either the frame shift form we just went through, or a truncating variant that stops right away, right after that nucleotide change. So you may see something quite similar to this on your family members' reports. Okay, but it is important, I think, to talk about another type of variation that does come up in our community and has posed for a lot of questions to all of us. And these types of variants are called missense variants. So instead of shortening the protein, like those last 2 variants that we just talked about, a missense variant causes a letter change, but doesn't shorten the protein. So this is a made-up variant. This variant doesn't exist, but it is a good example of what we would see for a missense variant, if that's what's listed in your family member's report. So in this case, the coding DNA change happened at 1297, and instead of an A, it became a C. And then in this case, when that letter change from an A to a C in the DNA resulted in a change in the amino acids in the protein from a lysine to a phenylalanine, but it doesn't shorten the protein. So it just changed that one amino acid. And these missense variants can absolutely still have a significant impact in how the gene works. It just depends on where they are and what they're doing to the gene. And in our community are a little bit harder for us to interpret at this point, but is certainly something that we've come across as well. Okay, so we're going to dive into the final section here is talking about ASXL variants. So what about our community that makes this a little bit more challenging? So this is kind of a pictogram of the ASXL genes. These are actually the proteins. And as you can see, the three proteins that are produced by the ASXL genes are quite similar. The different color areas are just the active domains where the protein does certain things, and they're a little bit different in each of the ASXL genes. But the biggest thing that you can see here is that the largest part of all three of these genes is what we call the last two exons. So each of those little dotted lines makes up the space in between each dotted line is an exon. And they're kind of just individual spaces within the gene. We talked about that picture I showed of the exome sequencing where there's exons, introns, exons, and that the exons are the active part in the DNA and the introns are sort of the intervening spaces. So in this case, the ASXL genes, just the way that they're structured, their last two exons are by far the biggest of all their exons, and their earlier exons are much smaller. So for ASXL 1 and 2, there are 12, it's exon 12 and 13. For ASXL 3, it's 11 and 12, so it has one less exon in ASXL 3. But those are certainly the largest portions of the gene, and therefore also the area where we see the most genetic changes. This is an example from one of our publications back in 2023, where we mapped out the genetic changes that we found in the ASXL community. In this case, again, you can see exon 12 and 13 are where all the variants are located. You can see on the top, these are the ones that were reported in our paper, and the ones on the bottom were other individuals that were reported in ClinVar, which is a database we use in genetics to kind of report genetic variations and have a tool for people to use it to interpret any changes they might find. So pretty clearly in ASXL1, we found that these changes are localized to the last two exons. And I know it's hard to tell on here, but if you actually were to look at each of these variants with your new set of tools here, you would see that all of them are truncating variants, either frame shift variants or sort of truncated right at the end. When we look at ASXL2 and ASXL3, we see a pretty similar pattern. So this one's not from a publication, it's from a program called Protein Paint, and it again is mapping variations from ClinVar. So these are what labs have called as disease causing. And the little lollipops that are in red or orange are truncating variants, and then the blue are missense variants. And these are all things that the lab has called as disease causing, either pathogenic or likely pathogenic. So you can see for ASXL2, the lab has primarily called truncating variants as being disease causing, and again, only in the last two exons. And then for ASXL3, the picture is quite similar, where we see truncating variants being called likely pathogenic and pathogenic. There are some that have sort of stretched outside of the last two exons. And again, this is one of the challenges in that these databases are relying on what labs are making calls, and all labs do not make the same calls. So much of this data is still sort of not tested and may not have had any functional data to back it up, but is based on what labs are calling as disease-causing changes. Okay, so what are some of our challenges then? So some of the challenges in the ASXL space have to do with the variants being inherited. So we've reported quite a few families that have unaffected parents that have passed on ASXL changes to their children. We have some parents who are mildly affected that have passed on changes to their children, and we have mosaic parents. And so Mosaic parents, the example here is of this heart up in the corner, where if you just took all those little pieces, they don't make up any special picture, but when you put them all together, they make a heart. So it's much like our bodies. It's common for us to actually have genetic changes that are present just in some cells in our body and not in every cell. So we are all naturally mosaics in that capacity, but sometimes an ASXL change can be mosaic and be present just in the parent's eggs or sperm cells, or just in a small percentage of the cells that you may find on blood testing or cheek swab. And so that always makes things a little bit more tricky as well. Oftentimes the mosaic parent may be unaffected, but can have a child that's affected if this ASXL change is present in all the cells of the body of the affected individual. So that certainly made for some challenges in understanding how to interpret these results. When these changes come from unaffected parents, it's possible that these may be benign changes that have no impact, and therefore they come from a parent, but it's not the answer for that child. It's also possible that those genetic variants, something happens when they get transferred from a parent to a child. And so a parent is not affected, but their child may be affected. And these are questions that we still have a long way to go to try and truly understand. It also adds to the challenge that some ASXL variants can be seen in healthy adults. So there is a phenomenon called clonal hematopoiesis, which means that in the red blood cells and the white blood cells of adults, as they age, they can acquire ASXL changes. So these are not ASXL changes they were born with, but something that happens in their blood cells as they get older. And when that happens, we can pick it up on genetic testing, but it is not the same as having a child that had an ASXL change present in all cells in their body when they were made. And so this can make it really tricky, because when we look at some of our control databases where we've sequenced healthy adults, we will find some of these genetic changes in ASXL genes that are the exact same variants as the kids that have ASXL-related disorders, but there is a different phenomenon and a different reason that the parents have, or the adults have, that in their bloodstream. So that makes it tricky to interpret some of those changes we find. The other challenge is that the clinical spectrum here is really broad and keeps getting broader. So there's absolutely a milder end of the spectrum that we see in ASXL disorders. And in that case, it can make it really tricky for us to draw conclusions, because it's perhaps that someone doesn't fit perfectly into the clinical box of what we think an ASXL disorder looks like, but perhaps they are just broadening of the spectrum and they are on the milder end of the condition. And so it makes it very difficult for us to look at the clinical picture and make a definitive call about whether or not their change is causing the condition or not. And that one of the biggest challenges we have still is limited functional studies. So we talked a little bit about functional studies being one of the tools we can use to understand a genetic variation. And so these are, again, studies that are often done on patient cell lines. So, either blood or cheek or skin samples, and studied in the laboratory setting to actually see what is the effect of that DNA change on the protein or some intermediate in the process. And those types of studies are really quite limited in ASXL. We have a methylation signature for ASXL1 only. And so we can use that to help interpret any variations we see in ASXL1, but the tools for ASXL2 and 3 are still being looked at. And so we don't always have a good way of understanding what someone's specific letter change in an ASXL gene might be doing specifically in their body. But it's certainly something we want to work on. Okay, so pulling it all together, covered a lot of ground, but a few big take-home points. Really important that you understand that variation in our DNA is normal. So it's normal to have genetic changes, and it's quite possible to get a genetic change reported on a genetic test that can absolutely be a normal or benign finding, and we just don't have enough information yet. So there's a lot that goes into the interpretation, and it can be really tricky for a lot of reasons, but I wrote a couple of them here, right? So humans are the persons who make the variant calls, so it's not a perfect science. Our databases of normal data are imperfect. We have limited functional data to actually investigate each person's letter changes, and we still don't understand many of these genes and many of their conditions fully. Variants of unknown significance are really common, and it takes time for us to resolve them. So we oftentimes have families that get stuck in this limbo space where we have a genetic change, we're not sure whether or not it's the answer, and honestly, we have to just wait and have more information over time. Wait for functional studies to develop, wait for technology to improve, wait for publications to happen where other people have that same genetic change. So it can take many years before we are able to resolve or fully understand some of these uncertain variants. And again, so that resolution process really occurs when we have more data, when we have other affected individuals, and we have better functional studies, and when we can really truly understand the disease processes better. So again, I encourage you to work with your geneticists or your clinicians and your treatment team to help interpret your specific results. And I always encourage families to follow up. It's really important. The data that we're learning, it's very fluid and it's changing. And so it's important that you have that long-term follow-up with your medical team and that you continue to reinterpret and look at these genetic changes over time. Again, many of our individuals who end up with uncertain variants may in fact have a different genetic condition that was not found on their testing, but as the technology improves, might be found in the future with better knowledge and technology. So it's important to keep looking if you have an uncertain result that we haven't been able to clarify, and also continue to follow that uncertain variant to see if it gets reclassified in the future. So I want to thank you for your attention. We covered a lot of ground here. I think we have a little bit of time to open it up to questions, if that's all right.